Hello, welcome to this video. This is Unzadani. So in this lecture, we are going to talk about the levels of protein structure. So we need to understand that protein structure is of three levels, which are primary, secondary, tertiary, and lastly but not the least, we have quaternary structure. Quaternary structure. We talked about these levels in the first lecture, but here we're going to go into details on each of them. So let's go with the first one. We start with the first one for primary structure. What exactly do we mean when we say primary structure of a protein? So the primary structure of a protein is just defined as the number of amino acids, number and the sequence of amino acids. So like we can have this amino acid, this is an amino acid, then the next one is an amino acid, then we have third one, in the fourth one, and the fifth one, so like this. So let's write it like this. So this is amino acid 1, then this is amino acid 2, this is amino acid 3, amino acid 4, amino acid 5. So this is the primary structure of a protein, since you just have the sequence and the number of amino acid in a polypeptide structure. So this one stabilized by peptide bonds. So these are peptide bonds 1, 2, 3, 4. So these are the peptide bonds that we're talking about, whereby this amino acid will make a peptide bond using the carboxyl group, then the next one will use the amino group, just like that. Carboxyl group, amino group, carboxyl group, amino group. So that's the primary structure of proteins. So let's go now to the second one, which is the secondary structure. secondary structure. So secondary structure, this is just the arrangement and coding of the amino acids in the polypeptide. So the coding and arrangement of amino acids in the polypeptide. So we have two secondary structures, two types of secondary structures. So the first one we have alpha helix. Alpha helix. So for alpha helix, let's try to draw it more like this. Like this. So then you can have hydrogen bonds in the structure. So here you just have, okay, let's say alpha helix. So here, so the secondary structures are stabilized by hydrogen bonds. So here you just have intra hydrogen bonds here, which are hydrogen bonds within the same structure, one structure. So that's alpha helix. So the alpha helix is spiral and it is right-handed. So an example of a protein that has alpha helix is keratin, which is a fibrous protein. Fibrous protein. So keratin is part of nails. Find it in the structure of nails, wool, hair, beaks, feathers, and bones. So that's why you find this one. So keratin is entirely alpha helix. So let's go to the next secondary structure, which is a beta pleated sheet. Beta pleated sheet. So this one can be represented by silk, fibrin, which is produced by silkworms. Silkworms. So this one contains polypeptides which are adjacent to one another. To each other. So this one, let's try to draw it. So for example, you have this one. Let's say this is the structure. So let's assume this is the structure. So then you have hydrogen bonds. So like at the end here, you have hydroxyl group, hydroxyl group, and on the other one you have this. So this is a hydrogen bond that is formed here. Let's say for example also here you have a hydrogen bond, also here you have a hydrogen bond, and at the far end you have a hydrogen bond. So since this one here, so you can have amino group here and H2 group, then here COOH group, also this one have COOH group at the end here, then here you have an amine group. So as you can see, an amino acid is ready from the N group towards the carboxyl group. That's how you read the polypeptide. So like this, so this will be the direction, then this one to be this direction. So as you can see, antiparallel, the polypeptides are antiparallel. So here, as you can see, the hydrogen bonds that are formed are called the interchain hydrogen bonds because they are formed between polypeptides that are adjacent to each other. It's not like in the alpha helix where the hydrogen bonds were formed using a single polypeptide. So interchain, I mean, not intra, interchain. Interchain hydrogen bonds. So let's go to the next level, which is the tertiary structure. So this one we already mentioned to say it is stabilized by hydrogen bonds, and hydrogen bonds are broken by heat. hydrogen bonds broken by heat. So let's go to the next level now. So the next one we have the tertiary structure. So the tertiary structure, this one, this is the formation of a 3D structure of a protein. This is the 3D structure of a protein. So here the interactions that stabilize this level of tertiary structure are, we have peptide bonds, then we have hydrogen bonds, then we have disulfide, bonds, and the hydrophobic interactions So as you can see, the peptide bond is a bond that is formed between amino acids, so in the sequence. Then hydrogen bonds, these arise because of the interactions between the side chains of the polypeptide. Hydrogen bonds are formed between amino acids that are polar. Then disulfide bonds are formed between amino acids that have got sulfur in them, like cysteine. When you have two cysteine reacting, they form disulfide bond. Then hydrophobic interactions, these are interactions that arise due to the non-polar amino acids that are close to one another. So these are just induced interactions between non-polar amino acids on the side chain. So the structure may be like this. Maybe something like this. Let's draw it. Yeah, something like this. Yeah, so like here, you can have these amino acids such as proline, these amino acids. So these, we also have this one CH3, this one CH3. So these are what we call hydrophobic interactions. Hydrophobic interactions. Then here we can have amino acids such as amino acid that has OH group here. Then we have another amino acid that may have something like this. Yeah, so here they form a hydrogen bond. So these are hydrogen bonds between amino acids that are polar. Then we have this. So these are disulfide. Between amino acids that have sulfur in their chain, their structure. So we also have amino acids that are charged. For example, we can have this one CH2. Then we have a 2 here. Let's say 4. Then NH3 plus. Then we have another one. Let's try it using a green. For example, this is amino acid. We have CH2. Then we have C like this. Then we have O, then it's so positive and negative interactions. These are ionic. Ionic interactions. So an example of a protein that has got a tertiary structure is myoglobin, which is that stores oxygen in the muscles. An example. Tertiary. Globular proteins. So globular proteins are myoglobin, myoglobin. So this one has got 153 to 154 amino acids. So let's go to the other. So the other example. Okay, let's go to the next level, which is the last level, which is quaternary. quaternary structure. So this one involves polypeptide chains. Or subunits. Coming together to form a functional protein. So these subunits are held together by peptide bonds. Ionic interactions. Disulfide. And hydrogen bonds. And hydrogen bonds. And also hydrophobic. Also hydrophobic interactions are also involved. For example, van der Waals forces. So hemoglobin is one example. An example. So this one has got subunits. 4 subunits. We have 2 alpha and beta subunits that interact. And 2 beta subunits held together by those interactions. So another example is insulin. So insulin, this one is held together by disulfide bond. By disulfide bonds. So in insulin we have chain A and chain B that interact with each other. So we can have like this one, something like this, and another one like this. So let's use a green color. So here, maybe joined here. So these are disulfide bonds. These are the disulfide bonds that we are talking about. So this is chain A. This is chain B. So let's look at the functions of now. So insulin regulates glucose levels in blood. In blood. So when there's too much glucose, insulin promote formation or synthesis of glycogen, which is the storage form of glucose. But when the level of glucose becomes low, this glycogen will be broken down in order to release the glucose. So let's look at the last one, collagen fiber, which is a fibrous protein. Collagen. So collagen is fibrous. This is a fibrous protein. So the primary structure has a lipido has a repeat of 3 amino acids. And these amino acids are glycine, proline, and another amino acid. And another amino acid. So in the secondary and tertiary structure, tertiary, so proline and glycine allow the polypeptide chain to be tightly coiled. Peptide to be wound into a tightly coiled structure. Which is left-handed helix. Which is left-handed helix. So another thing